Welcome back. In the last video, we saw how alpha oxidation of a 12 carbon fatty acid ultimately produced adipate. And so right here, I have drawn the structure of adipate. And right here, I want to explore how adipate ends up turning into things that we can actually use in the cell. And I think what you'll find is that really in this degradation process, we're not really learning anything new. We're actually going to use a form of beta oxidation. Okay. So initially, what's going to happen is you're going to use an enzyme called Adipyl-CoA synthetase. And actually, this enzyme is functionally identical to the fatty acyl-CoA synthetase, except for this one is specific. It is specific for something that has a carboxyl group. Or you could view it on this end if you wanted to, but it has a terminal carboxyl group, so it's specific for that. So you're going to put in an ATP, right? You're also going to put in a coenzyme A, right? And it's going to kick off AMP and pyrophosphate, right? It's going to kick off pyrophosphate. Of course, a pyrophosphatase drives the reaction forward with the release of free energy. And ultimately, what you end up with is with this. So 1, 2, 3, 4, 5, 6. You end up with this right here. So this, we call this Adipyl-CoA. And now the enzymes that oxidize this are specific for two things. Number one, they're specific for the coenzyme A, right? They're specific for that, but they're also specific for this carboxylic acid at the end. So the enzymes that do this are different than the normal beta oxidation enzymes, although the mechanisms are virtually identical. So in this next reaction, essentially what we're going to do is we're just going to use an FAD-dependent dehydrogenase. And so what we're going to end up with is we're going to end up with FADH2, right? And like I said, it's more of an exercise in beta oxidation. So we're going to find that it's virtually identical, except for the fact that the substrate's different. So what we're going to end up with is an enoyl-CoA. So 1, 2, 3, 4, 5, 6, right? We're going to end up with an enoyl-CoA. And then that's going to be consumed also. It's going to be consumed by an enoyl-CoA hydratase. Enoyl-CoA hydratase. And of course, that's where you have water coming into the reaction, and it's basically going to make something that we've seen something very similar to. It's going to make a beta hydroxy acyl-CoA, right? Then you're going to have an NAD-dependent dehydrogenase, right? So this hydroxyl group at the beta position, right? This is the beta position. Here's the alpha, here's the beta. The hydroxyl group at the beta position is going to be dehydrogenated. And so you're going to end up with an NADH. And so what you end up with is this. You end up with a beta keto acyl-CoA. Make sure 1, 2, 3, 4, 5, 6. Yep. So we have a beta keto acyl-CoA, right? Well, then we're going to have a reaction that we've seen many, many times. And so what I'm going to do is I'm going to do this in green. And so we're going to have a coenzyme A. And recall that the coenzyme A has that lone pair on the thiol. And essentially what's going to happen is the lone pair is going to come over and do a nucleophilic attack on this carbon. The pi bond's going to kick up. Pi bond's going to kick back down. And this guy's going to come out. Well, actually, excuse me. Let me regroup. Sorry, I made a mistake. It's actually not that carbon that it hits. Excuse me. It's actually not that carbon. It's actually this carbon. So the pi bond kicks up, kicks back down, and then this bond comes out and abstracts a proton. And so what you end up with is two things that we know. You end up with a succinyl-CoA, right? You end up with a succinyl-CoA, right? And of course that will go into the TCA cycle, right? But you also end up with an acetyl-CoA. you also end up with an acetyl-CoA, right? And of course, that goes into the TCA cycle. Of course, they enter at different enzymes. Succinyl-CoA is going to get consumed by succinyl coenzyme A synthetase. Acetyl-CoA is going to get consumed by citrate synthase. So what have we seen in this pathway? Well, in omega oxidation, we generated adipate. But just like, I mentioned in other videos, our body doesn't like to waste things. Everything there is in our body, there's some use for, and usually there's some catabolic pathway for. Adipate is no exception. So like we see, we're doing a pathway that's virtually identical to beta oxidation, except for the fact that it's specific. The enzymes are specific for this carboxylate right here. So there's a fatty acyl-CoA synthetase, an FAD-dependent dehydrogenase, an enoyl-CoA hydratase, an NAD-dependent dehydrogenase, and a thiolase. So that's what this enzyme is. This is a thiolase, and it's going to hit this carbon, do a nucleophilic acyl substitution, and essentially it's going to kick off acetyl-CoA. Okay, so this has been the pathway on adipate metabolism, and I hope you enjoyed this video. In the next video, what we'll end up doing is we'll look at alpha oxidation, and alpha oxidation is probably the most difficult. In terms of understanding, it's the most difficult oxidation, so we save it for last. And a lot of times they don't even do it in Biochem 1, but we're going to do it. We're going to do it anyways. And then after that, there's going to be a playlist on ketone body function. So hope to see you there.